Actually, tocotrienol is actually a form of vitamin E. Vitamin E consists of two different series or classes. One called tocopherols and the other one tocotrienols. Actually, they have very similar chemical structure of a head and a tail. So the difference between the two actually lies in the tail. For tocopherols, the tail is fully saturated. For tocotrienols, you have 3 double bonds at the 3, 7, and 11 positions. So, all this while, if you were to buy a vitamin E product in the market, you only get tocopherols, and usually only one, alpha tocopherol. Why you don't get tocotrienols is because in the past, tocotrienols are not available commercially. It's difficult to find in nature until we found plenty on palm oil, and therefore Malaysian companies became the first to extract and commercialize. And because of this now, there are availability, there are more products in the market now having tocotrienols. Also research, because in the past also it's difficult to find tocotrienols for research. And if you find that in recent years there's a lot of research done on tocotrienols. And what they found, very interestingly, tocotrienols have some unique activities not found with the tocopherols. And one of those that has actually caused a lot of excitation to the scientific community is actually on the neuroprotective activity. It all started in Ohio State University, where they found that it can prevent nerve cells under culture from dying when challenged with glutamate. And glutamate is the toxic chemical signal released during a stroke. Then they went on to do on rats, induced stroke in rats. So with and without tocotrienol, they found that those given tocotrienol, when they induce a stroke, the damage is smaller than those without tocotrienols. Then they went further to do on dogs again. Similarly, they found that those given tocotrienols, if you induce stroke in them, the damage is much smaller, significantly smaller than those on not given tocotrienols. Here we have done a study in humans itself, people with what we call white matter lesions. Now what are white matter lesions? You see, our brain, you need a lot of energy, and therefore the brain is supplied by a massive network of blood vessels. But unfortunately, these blood vessels can get damaged. For example, aging itself and small blood vessel disease, they call it, cause damage to the small blood vessels. So when these blood vessels are damaged, the blood supply is disrupted. So when the blood supply is disrupted, it's just like in a stroke situation, you don't get blood flowing to the brain, and they're starved. And then these nerve fibers in the brain will die off. And from an MRI, you see the white matter, they call it white matter lesions. These are lesions in the brain. Basically, these are nerve fibers degenerating and dying off. So once people have white matter lesion, then they will start to progress. And they get more and more and more. So what we did is, we have two groups, one given tocotrienols, one placebo. Study was double-blind, we don't know who is given what. Even the volunteers, they won't know what they have been given. So we MRI at baseline, 1 year and 2 years. And then we see how the lesions grow. And very interestingly, those on tocotrienols, on average, we find that the lesions are stabilized. Those on placebo, they start to grow bigger and bigger, like what has been reported by other researchers. So based on that, it proves this is the first human study to show that the tocotrienols indeed can protect your brain cells. Now, white matter lesion, certainly the world scientific community, a lot of neurologists are so excited about white matter lesion because today they realize that white matter lesions are associated with many neurodegenerative disorders. For example, dementia, Alzheimer's disease, and a lot of other neurodegenerative diseases. So, based on what we have obtained so far, tocotrienol have immense potential, for example, in preventing further degeneration of your nerve cells in the brain, and therefore has a potential of preventing or delaying development of cognitive impairment, dementia, and even Alzheimer's disease. Now in my lab, I have this little worm called C.elegans, and I have a transgenic version of it, where the human genes have been, or disease gene has been inserted into the worm. So what this worm will do is, when we induce them, they will start to produce this beta-amyloid that is found in Alzheimer's disease. And once they have this beta-amyloid accumulated in the body, they go into paralysis. So what we did, we have a control, and we have a group where we give them actually tocotrienols in the culture. Then we see the time they get into paralysis under the microscope. And we found that with the tocotrienols, the time they go into paralysis is much delayed. So there's a lot of potential there. Latest what we've done is that we look at the whole transcriptome. Basically, we look at the whole genome of the worm, see how the different genes are activated, so-called up-regulated or down-regulated. We want to see what happens in the genome, whether tocotrienols suppress the genes that produce the beta-amyloid, or other factors coming in and all that, you see. So we are waiting for the results. Hopefully I get it in 1 to 2 months' time. That'll be interesting.